For a conversation about the next frontier of immunotherapy, please welcome Brian Czerniecki, Chair of the Moffitt Cancer Center, Nina Bhardwaj, Director of Immunotherapy at Mount Sinai's Icahn School of Medicine, and Jedd Wolchok, Chief of the Melanoma and Immunotherapeutics Service at Memorial Sloan Kettering. Here to lead the conversation is author Charles Graeber.
All right, so just kicking this right off, you wouldn't guess it from something like this, but cancer immunotherapy was once the most underfunded and overlooked direction for new therapies, and that changed recently and in a really profound way, quite obviously, and that's created incredible new opportunities, a lot more funding, and an imperative for industry and academia and every branch possible to join this fight and work together in a collaborative fashion. That's also created new challenges that you've never had to deal with before, when you were just scrapping for obscure research dollars. Can you speak about some of that? It's been an issue in the news that we'd like to take care of.
Yeah, so Charlie, thank you very much for the question. I think what you're really talking about is the importance of transparency in medical research in terms of financial conflicts of interest. And I think this is a very, very important area. I've always tried very hard to fully disclose what's relevant to the topic, and I think many colleagues do as well. But this is a very hard thing to do across all of medical and scientific research because different journals have different requirements from each other, and even sometimes there are inconsistencies within a journal about what needs to be disclosed. So I think we all agree that transparency is very, very important, but I think all of the stakeholders in this area really need to come together to develop some uniform guidelines for what is a meaningful amount of disclosure so we can move ahead in a very transparent way.
And getting right to what we're moving ahead with. What's most interesting, there's a gee whiz factor that we get to talk about. This is a relatively short amount of time that we have to discuss it, and there's so much, not just on the horizon and that old horizon of 20 years hence, but now they're talking about over 3,000 new therapies in the pipeline. I'd just like to really open this up to the entire panel, starting with you, Nina, I guess. What's exciting? What's next? Where should we be looking for the next breakthroughs? What are reasonable expectations for them?
I think what's terribly exciting is obviously approval of many checkpoint inhibitors, antibodies that reverse immune suppression in the tumor microenvironment, and which have made dramatic impact in the treatment of so many patients with cancer, in particular melanoma is a great example. So that's a wonderful platform, and that's been obviously celebrated this year with the awarding of the Nobel Prize to James Allison and Dr. Hanja. And now the challenge is beyond that, and we're incredibly exciting field because we have so many combinations now to test in addition to these checkpoint inhibitors, and they include other checkpoints. They include oncolytic viruses, the use of virus injection directly into tumors, a new platform of tumor vaccines that's being developed as a personalized therapy. And so I think the challenges are enormous. It's very exciting, and we'll see where we go. But as I think Jedd was alluding to earlier, we follow the data. We see how our patients do and follow the data.
We were speaking earlier about when this was a field where people struggled really to make work. Before it was agreed upon that the immune system could recognize cancer, could recognize tumor cells, and could kill those cells, and yet you could help them do that. There were glimmers, right? There were glimmers in the darkness. Can you talk about how important those glimmers are, how the exceptional patients sometimes lead to important understanding, something outside of the larger pool, and what glimmers you see now? Starting with you, Brian, I guess.
Yeah, in the past, we've seen patients that have documented cancer that have, for one reason or another, spontaneously cured themselves or lived well beyond where they were supposed to. And I do remember patients that I had seen down at the National Cancer Institute, which had gastric cancer that, miraculously, either through getting an infection or some other reason, triggered their immune response, and they became cured of cancer. The problem was it was very few and sporadic. And we see the same thing in today's day and age, with sometimes a therapy treating a local tumor, causing other tumors to disappear. And I think we're getting to a point in the immune system where we're understanding that, so that it's happening more and more commonly than the infrequent patient.
Mm. Do you have anything to add to that?
I really echo what both Brian and Nina had said, and I think Nina and I actually shared a mentor, Dr. Lloyd Old, who's really regarded as one of the parent figures in the field, who really encouraged us to study individual people and the course of their disease very deeply. And there were glimmers, and I would actually point to an immunotherapy that's not spoken about very much anymore, an immune hormone called interleukin-2, which was actually developed by the blood, sweat, and tears of Dr. Steve Rosenberg at the National Cancer Institute, which really cured a small number of people with melanoma and kidney cancer. And while the number was very small and the side effects of that therapy were considerable, the fact that a medicine that did nothing directly to the cancer cell could cause long-term, decades-plus remissions in anybody, for me, was an indication that we needed to understand more about what regulates the immune system, why some tumors could be recognized, why others are sort of cloaked molecularly from the immune system, and how that basic science knowledge, which was accumulating and finally rewarded with the Nobel Prize this year, could lead to therapies that have transformed the lives of some patients with some cancers.
Could we, in terms of what's next, or what we might expect to make an impact next, in addition to the combinations, and you hit upon some of that. Vaccines. People ask about vaccines quite a bit. Could you explain how a cancer vaccine would work in broad terms, and how realistic it is for us to think that such a vaccine could work in the future when it didn't in the past?
So many of us, Jedd and Brian and myself, have been working on cancer vaccines for a long time. And of course, we have ones that prevent cancer, such as those for HPV-associated cancers and HBV-associated cancers. But clearly, there's a need to bring this into the therapeutic setting. And really, there's just one approved vaccine broadly in this country, and that's for prostate cancer. So the need is great, and the field, where the field is going now is to try to actually sequence a patient's individual tumor and identify mutations within the tumor that lead to the production of novel proteins. And those proteins can be pooled together to develop vaccines for a patient. So the field is currently looking at preparing vaccines that will be personalized in one sense, but also identifying mutations that are shared amongst patients and can be incorporated into vaccines. And we call those neoantigen vaccines. And there are a number of studies now that are looking promising. I would say it's very early still, but hopefully with, again, combination therapy may allow patients to further boost their immune response. And then these mutations are being incorporated into many different vaccine platforms. And that's another area of research that all of us are looking at very carefully. What is the best vaccine platform to use? So we'll see more of that coming in the next decade.
Great. Did you have something to add on that?
Yeah, I'd like to expand on that. It was a really good description, thanks Nina. And I think it speaks to a little bit of the disappointment that happened in the past when vaccines were given by themselves as a therapy for cancer. And now we have the ability to amplify the immune response to the vaccine with some of the other immune modulating medicines, these checkpoint blocking medicines that perhaps would allow the induced immune response from the vaccine to be of a greater magnitude and more long lasting. But as someone who's been in the field for over 2 decades, I think it's extraordinary to think about what Nina just said and how it was probably considered science fiction until quite recently, that within a few weeks or a month or two, we could go from taking the gene sequence of a person's tumor to preparing an individual vaccine within that time frame seemed completely out of reach just a few years ago. And while there are still many challenges in terms of manufacturing and the computational aspect of it, of selecting the best parts of the mutated genome to target with the vaccine, the progress, in my opinion, has been extraordinary and really reflects a deep commitment to the science that this field has made.
We talk about T cells, if we talk about anything specific at all. It's the T cell as hero. Could we talk about other cells?
Yes.
Other heroes that we're now recognizing.
B cells?
Right, so, whether it be, natural killers or I'm thinking specifically of dendrites and the like.
Yeah.
Could you talk about some of the other cells that we're now recognizing?
Sure. One of the immune therapies that's never discussed really as an immune therapy is the use of antibodies. So Herceptin, an antibody that targets the protein HER2 on some types of breast cancer cells, it actually has been touted as a targeted therapy, but it really works too because it's an immune therapy activating the cellular component of the immune response. And the B cell arm, I think people are starting to realize can be an important player with the T cell arm of the immune response, and you're seeing more and more investigations going on looking at how B cells and T cells interact at the tumor site to make a more effective immune response.
Any others? I'm always interested in players.
We also work on other immune cells called dendritic cells and also natural killer cells. These are white cells that are components of the innate immune system. And a lot of work that we're doing currently is trying to harness the ability of dendritic cells, which are the sentinels that capture tumor antigens and pieces of tumor to prime or activate a T cell. Those are being used as a platform, a vaccine platform, and NK cells are also being harnessed through expansion and molecular manipulation and being delivered back to patients, as well as another way to introduce an effective anti-tumor response.
And I'm going to get to we're going to get to some questions from the audience in just about a minute or two, so you might want to put those together. But before we do that, I know that this is 100 years in 25 minutes, and I know as soon as it's over, you're running straight to the clinic, for example. And can you talk about what it looks like in the clinic now? What your waiting room looks like?
Yeah. So I think this is a very good reminder of how far the field has come. So my clinical practice is mainly in melanoma, and the nature of the waiting room and outside my office is very different now than it was 7 or 8 years ago. And I think melanoma in some cases is a bit of an extreme example of how these medicines have impacted different cancers, but I think within my professional lifespan, I've seen the median survival of people with melanoma change from 6.5 months to over 4 years. And that was in a pretty short period of time. And so it used to be very difficult for me to see the same people in the waiting room at the same time, but now I see old friends in the waiting room. And I think that is not only, of course, very rewarding for us, but the spirit of hope that is there among the people who have just been given a devastating diagnosis is infectious. I just wanted to abuse the privilege of you asking me and add one little comment. First of all, I wanted to congratulate you on your book, Charlie.
Thanks.
And say that there are two other authors of books about the field in the room. Your last question about other cells reminded me of a book that Stephen Hall wrote when I first entered the field in the mid-90s called A Commotion in the Blood. And one of the chapters was actually entitled The Rise of the T-Cell Chauvinists. And so I think that we are still a bit chauvinistic, but I think we really need to dig quite deeply into what these other cell populations and products are so that we can provide more potent answers.
Excellent. Can we open this up to questions? Because we've got about 5 minutes, and I can't see out there, but we probably have. There's somebody with a microphone.
Great. This is Michael Strand at Immunorestoration. Look straight ahead, Brian. The concept of vaccine platforms has been discussed, and I wanted to know if you could tell us a little bit about the platform that you've developed using dendritic cells and T cells, and what your success rate has been, and where you are taking that particular platform in the future.
So similar to what Nina mentioned, we use dendritic cells that are activated rapidly that we get from a patient's peripheral blood, and we activate them out of the body, give them a group of signals, and then infuse them back in various ways into patients. We do it in early breast cancer, but now we do it prior to chemotherapy to try to get an immune response in the tumor to make our other therapies better. We have several trials ongoing. With all immunotherapies, the earlier you use them in cancer, the more effective they are alone. But there's still good role for vaccines and in the setting of other therapies, combination, some of the checkpoint products, chemotherapy. And so the whole role of vaccines, I think, is changing as we learn more about the immune response.
I can't see anything, so these will be just voices from outside.
Gordon Earl, Albert Einstein College of Medicine. But I'm here as a step-parent who lost their son to melanoma after being treated with immunotherapy. And I understand the message of hope very profoundly, but there were many procedures taken over a 2-year period. And looking back, especially in a tragic situation, there are many what-ifs. What if this? What if on the surgery, on the treatment? What advice do you have to parents looking back? And we want the situation of hope, not despair. But if it didn't work out today, what do you say to dealing with the what-ifs, which everyone has to face?
First of all, I'm very sorry for your loss, and I think it's an important point that even though many people are helped with melanoma, it's not everybody, and I think it's a clear path for the need for more work. When I speak to the people in my practice about making decisions, I counsel them that we make the best decision that we can in the moment. There is no looking back, because if there were a right answer at that time, we wouldn't really be making a decision. We would all know what the right thing to do is. So I think in the absence of a clear gold standard of what the right thing to do is, all we can do is choose a path that seems right to everybody in the room in the moment. And I know that sounds quite a bit easier to say than it is to do, but in the absence of a uniform solution for everybody, I think that's where we are.
We have probably time for one or two more.
Hi, I'm Casey Smadzinski. I'm with NBC, but I'm here as a caregiver and patient advocate. And first, thank you for all of you for being in this space and trying to advance the research. But my question is around looking at unpublished trials and research that seems like, as a caregiver, we're following a lot of trials and seeing when the trials finish and waiting for that therapy research to be released so that we can inform our person that we're helping to see what the path forward would be. A lot of trials, we celebrate the successes, but the failures don't become published, or they were published years later, where it seems like people today, and I know there's conflict of interest between competing companies and they have their own interests, but what's your opinion on that? And should this research, that if we're going to celebrate the successes, should we learn from the failures in a quicker way?
Who wants to take the role of trials published and otherwise?
I think it's a critical point you raise. We often don't publish them, but now the clinicaltrials.gov and the government is actually requiring you to put online your results of your trials in real time when they're done, and I think that's important. But those don't get looked at by a lot of other researchers, and so we're incented for success, we're not incented for failures, and that's something that probably needs to be looked at and changed going forward.
Believe it or not, we are out of time. I'd like to thank our panelists and thank everyone for coming.